Deep inside the exocrine glands, a microscopic environment of healthy acinar cells operates in perfect homeostasis. But under specific conditions, that stability collapses into an aggressive, localized war zone driven by cascading stress signals. Historically, clinical consensus defined Sjogren's disease as a chronic autoimmune exocrinopathy characterized by dry eyes and a dry mouth. It was categorized primarily as a B-cell-driven destruction of the salivary and lacrimal glands. In this model, the epithelial cells were cast as passive targets. They were structural bystanders, destroyed by an unprovoked invasion of infiltrating lymphocytes. Modern single-cell transcriptomics and spatial omics provide evidence that contradicts this linear model. These high-resolution tools reveal a complex, three-dimensional network where multiple cell types are physically and chemically interconnected. Sjogren's is a dynamically organized epithelial stromal immune ecosystem. Lymphocytes interact closely with local fibroblasts, plasmacytoid dendritic cells, and, most importantly, the epithelial cells themselves. Epithelial cells function as immunologically competent initiators. They demonstrate dysfunction before the arrival of infiltrating lymphocytes, actively orchestrating the immune activation that leads to their own destruction. Disease initiation relies on a distinct polygenic autoimmune predisposition. The strongest baseline risk is conferred by specific HLA alleles, such as HLA-DQB1 and HLA-DRB1, which facilitate aberrant antigen presentation. This animated genetic diagram details a secondary layer of risk, non-HLA susceptibility genes. Loci containing IRF5, STAT4, and TNF-SF13B predispose the patient to defective immune tolerance and a heightened interferon response. This genetic architecture can remain dormant until an environmental trigger is introduced. Evidence points toward viral exposures, specifically Epstein-Barr virus, which can act as a catalyst for these latent susceptibilities. The initiation sequence begins with a precise biological collision. A viral particle docks on a genetically primed host cell, injecting viral nucleic acids directly into the cellular space. Those nucleic acids interact with internal toll-like receptors, specifically TLR3, TLR7, and TLR9. These sensors are concentrated heavily inside the epithelial cells and localized plasmacytoid dendritic cells. The combination of polygenic susceptibility and viral mimicry primes the glandular environment for an explosive, self-directed immune response. That receptor contact drives the early molecular event known as epithelial interferon licensing. Activated plasmacytoid dendritic cells and epithelial cells respond to the viral presence by synthesizing and releasing a massive wave of type 1 interferon, primarily IFN-alpha. This diagram outlines the resulting JAK-STAT signaling cascade. The newly released IFN-alpha particle approaches the target cell and docks into the IFNAR receptor. That binding action drives an immediate intracellular phosphorylation step, activating JAK1 and TYK2 directly beneath the membrane. The energy from kinase activation phosphorylates STAT1 and STAT2. These structures capture IRF9 and unify into the ISGF3 complex. The complex breaches the nuclear boundary and binds to the cell's DNA, initiating the transcription of interferon-stimulated genes, or ISGs. The nucleus floods the surrounding tissue with mediators, including BAFF, CXCL10, and CXCL13. This creates the interferon signature observed in Sjogren's patients. This licensing process transforms the epithelial cell. It is no longer a standard exocrine unit. It has been reprogrammed into an active immune director. While the cell projects signaling proteins outward, a metabolic crisis occurs within its interior structure. Inside the salivary gland epithelial cell, the mitochondria begin to fail. High-intensity immune synthesis forces massive oxidative stress, leading to a depletion of cellular ATP. To survive the energy deficit, the cell undergoes a glycolytic shift. It alters its fatty acid metabolism and begins accumulating lactate in the cytoplasm. This 3D model reveals the structural consequence of that internal distress. The collapsing metabolism triggers an external architectural change, driving the upregulation of MHC class II molecules through the outer membrane. This immunometabolic phenotype allows the cell to function as a non-professional antigen-presenting cell. It holds autoantigens outward, actively attracting the surrounding immune system. Moving outward into the glandular stroma, we find local fibroblasts responding to these reprogrammed epithelial cells. Spatial transcriptomics show that these local fibroblasts act as active immune regulators. They are central organizers of tissue-level immunity. These fibroblasts secrete a specific cocktail of chemokines, CXCL13, CCL19, and CCL21, to recruit circulating lymphocytes directly into the glandular tissue. This spatial map visualizes that chemical pull. The chemokines haul lymphocytes into tight physical formations, constructing tertiary lymphoid structures within the tissue matrix. These structures are highly organized. CXCL13 pulls B cells together to form a dense inner follicle, while CCL19 recruits a distinct outer ring of T cells. The stroma has built ectopic germinal centers inside the gland. This architecture provides a permanent, fortified base of operations for chronic autoimmunity. Inside these structures, adaptive immune dysregulation takes over. B-cell hyperactivity drives tissue destruction, fueled by the BAFF to BAFF receptor signaling axis. The tissue environment is saturated with BAFF, a survival factor. It prevents the natural death of autoreactive B cells and drives them to produce autoantibodies, including anti-SSA/Rho and rheumatoid factor. This split-screen schematic tracks the simultaneous mechanisms of adaptive dysregulation. As B cells flood the system with autoantibodies, Th1 and Th17 cells lock on to the local tissue, blasting it with cytokines like interferon gamma and IL-17. These cytokines splinter the cellular matrix. They mediate direct epithelial injury, shredding the local architecture and releasing fresh autoantigens into the environment. T follicular helper cells maintain the germinal centers, while regulatory T cells fail to suppress the mounting damage. This circular infographic maps the resulting pathogenesis. The system functions as a closed, self-amplifying cytokine loop. Type 1 interferon drives B-cell recruitment, which creates autoantibodies and immune complexes. This triggers complement activation and Th17 polarization, causing epithelial injury that releases more autoantigens, which further drives interferon production. The initial viral trigger becomes irrelevant as the pathogenesis powers itself, trapping the local tissue inside a chronic inflammatory vortex. This mechanism carries a severe clinical endpoint. For approximately 25% of patients, relentless antigenic stimulation and BAFF-mediated survival drive the tissue toward lymphogenesis. Management focuses on specific clinical risk factors indicating this progression, such as persistent parotid enlargement, cryoglobulinemia, and depressed complement levels. When the disease breaks parameters, the tertiary lymphoid structures mutate. B cells undergo an aggressive monoclonal expansion, resulting in the development of extranodal marginal zone lymphomas. Because we can now trace these biologically programmable pathways, broad immunosuppression is no longer the standard. This dashboard illustrates molecular endotype stratification. Patients are categorized by their dominant biological signature, IFN high, B cell high, Th17 dominant, or metabolic dysfunction. Targeted therapies address these specific signatures. Anifrolumab blocks interferon signaling, ianalumab executes B-cell depletion, and telitacicept provides dual BAFF and APRIL inhibition. Research using patient-derived salivary gland organoids demonstrates that these mechanisms are biologically actionable. Even the metabolic collapse within the epithelial cell is partially reversible. Effective precision therapy for Sjogren's disease requires the simultaneous management of epithelial biology, stromal remodeling, and immune dysregulation.